Kevin, you can say it. I can tap it.
Oh.
First name. I have your first name. What's your last name?
Waters.
Waters. Waters.
Yeah.
Okay. Which institution you're from?
NYU.
NYU. Great. Okay. So you have any email we can use to contact you? Send you any information?
Yes, I can. Let me see. Let me put my email right now in the chat.
Okay. Oh, actually, I saw you're typing your name there. So
yeah,
so
I type it in. Okay, here we go.
Okay, gotcha.
That's perfect. My email.
Yep. And then Kevin, whose lab are you in?
Who lab I'm in? Do you know Mr. Sparks?
Mr. Sparks? Mm-hmm. No.
Okay, so Mr. Sparks, he speaks like he's a very good teacher. Something he teaches like a lot about Dominican people and a lot of rat culture. So that's what we get into with his class.
Okay. Do you have that gone, or do you want him to type anything else in?
No, Kevin already gave us everything, so that's yeah.
Yeah, so Kevin, as Brian alluded to, the company is a specialization in synthetic oligonucleotides, which are short DNA strands. And then the application for my tags, what we do with that product is you can target that product just like what Brian does with his baits. The my tags is actually a fish product or an in situ hybridization product. So what you do is you have this tube of or a pool, we call it, of unique oligos that will target a specific site, whether it's a genomic target, genes within the genome, or whether we're looking to target mRNA expression within cells. So these oligonucleotides have a little tag on them that's a fluorescent molecule, or it can be a non-fluorescent molecule. But that little tag allows you to visualize where these oligonucleotides are hybridizing, either within the chromosome or within the cell. So the MyTags product is more of a localization and functional product. We look at upregulation, downregulation of gene expression, or we look at maybe how translocation of genes across different chromosomes are happening. So with Brian, you're looking at sequences of oligos or sequences of base pairs. With MyTags, you're actually looking at localization of genes or gene expression products. So if you're using a microscope to look at your research, the MyTags product is what you're going to be using.
That's cool.
It makes really cool pictures.
Yeah.
I have a question for you real quick. So I want to know, so what would you do if when you okay, he said you would go?
What's the question?
The question is, I was asking, what would you do if when you okay, so he said, yes, we'll go?
I'm not sure I know what that is in reference to. Like who says
I'm
talking
about like the molecules of a chromosome to X and Y. Yeah, that like I was asking, like, what would you do if when you okay, so he said, yes, we'll go? Like in the same reference of the molecules.
Yeah, I guess we did not. What you say? Okay to go to who said that? Okay to go and.
Yeah, so I'll say it again because I think I didn't hear me. So I'll make sure I'm loud enough for you guys. I was asking, what would you do if when you okay, so he said yes, we'll go? Did you hear that?
No, you said okay and said
go,
right?
Do y'all want me to type it for y'all? I'll type it for y'all because I don't think.
Type it, please.
Okay, I'll type it. Okay, I'll type it.
Yeah. So when you're typing, Kevin, can you put your faculty's name there?
Yeah, I'm sorry. If you don't know, y'all, I'm so sorry, but English is not my first language. So if I do kind of feel confused, I'm so sorry. But please,
you're perfect. Okay.
It's not my first language. I'm Dominican.
It's going through perfectly. I just don't know if I know the context of what you're asking. So it's kind of on me to try to figure out what this is.
Okay, I got you right now.
Yeah.
All right, you can read that out now.
Yeah. Yeah, Kevin, it's if you okay, so he said yes would go. So who's you? Who is you? I mean, basically we're trying to figure out.
I'm Kevin.
Okay. And if when you okay, he said yes would go. Who is he?
The chromosome.
Chromosome. So the
And what do you want to do with the chromosome?
I'm pretty much trying to build a rat.
Okay. So you want to do like genetic engineering, like
Yeah.
crosses and stuff like that?
See, that's what I was saying.
Oh.
So who yes, you see yes will go. You see, it's like it's the conclusion of the chromosome of trying to build a genetically rat, you know, in a way of test rats, you know, like you know how we lab test, you know, like stuff like that, you know.
Is that something tags could do, Linda? Like if you're doing a cross and you know there's certain genes.
Well,
that
yeah,
are
I mean, you
one
can
and
look
not the
at
other?
different hybrids that are generated by crossing
Yeah,
different
definitely.
species, right, Kevin?
Right.
What else, you know what? If you go to the link here, which is I'm going to copy it again, Control C, Control V. If you go to this link, that's going to take you to our page that has all our product information. And if you look under there, there's different application sheets. So
I'm looking.
So I'm just going to jump into like if we're doing, you know, these genetic hybrids and crosses, like what's the goal? Like, are you trying to look for something to use in an assay? Or are you trying to figure out some sort of genetic feature? Like what's the, you know, when you do the crosses, what phenotype or effect are you looking for afterwards, like when you're done?
Okay, so pretty much, if you guys don't know, I'm pretty much doing a project right now. And with this being said, pretty much just tensors have been chosen out of my class to pretty much do projection of what we've learned with photosynthesis with when it comes to molecules and chromosomes in that area. So pretty much the animal I chose was rats because I wanted to pretty much learn more about because rats they're not really understood. Rats have flexible bones. Rats are really, they multiply. Rats run through the city like it's nothing. I be seeing rats outside pick up cheese pizzas. They be wearing Timberlands. They be having Nike sneakers. Yeah, so I just wanted to study rats. And honestly, I think, I have been way more obsessed with this project more than what it actually is. So it's to the point where I think I want to build my own rat community.
Okay.
Because I'm Dominican.
So we can't help you with the building of it, but we can help you with like the genetic testing.
analysis, yeah.
And
Y'all don't have a building with it? Y'all can help me with building it?
No, we would be the ones who, like, if you knew something on a chromosome, we could have it light up or not light up. Like, let's say you put in a new gene. We can give you an assay that would, like, look for that gene to make sure it's there, because that's one of the things you want to do when you do any crosses is you want to check the offspring to see if what their genetics are. Like, if you have, like, all these different things, once you cross them, sometimes they get lost during the, meiosis and mitosis.
Right, yeah. Okay. This is that right? Nope.
Like that ass shake for you? I'm gonna shake the ass all for you, Linda. You want this black ass? I'm gonna give it to you, you white old bitch. Take this black booty. You want to smack it. Smack this.
Turn this off.
Where the fuck she went? Tell that white bitch come back before I beat her ass. I'm left with you now. Period. Hi, scientists. Why the fuck she ran up out of here like that? What? Mr. G-Wang, what the fuck? Y'all niggas just left me in here by myself? Girl. Why they all hit it? Why the